Hello students, welcome to Chargaffs classes. Today our topic is biomolecules and I'm going to discuss about deoxyribonucleic acid, that is commonly known as DNA. And I'm going to discuss some of its features, how it is formed, and I'm going to discuss this through some points. So now let's get started. The first point about DNA is DNA of mitochondria and chloroplasts are not associated with histones. So, DNA of mitochondria and chloroplasts is not associated with histones. Now the next point is, so in the first point, the DNA of mitochondria and chloroplasts is not associated with histones. Now the next point is, in a deoxyribonucleic acid or DNA molecule, amount of purines is equals to amount of pyrimidine. So in a DNA molecule, the amount of purine is equals to amount of pyrimidines. So from this we can say that A plus T, that is equals to G plus C. Now A, that is adenine, T, that is thymine, G is guanine, and C, that is cytosine. These are the all four elements of DNA. So in DNA molecules, amount of purine is equals to amount of pyrimidine, and from this we can say that the total amount of adenine and thymine is equals to the total amount of guanine plus cytosine. Now it is called the Chargaffs equivalence rule. This is known as Chargaffs equivalence rule. That was in 1950. Now the next point is, and is third one, that is the ratio of adenine plus thymine and guanine plus cytosine is constant in any species. In any species. So it doesn't matter what kind of species it is, but always this ratio adenine plus thymine and guanine plus cytosine will remain constant. Now the next one is DNA is made up of two long chains, that is strands. So DNA is made up of two long chains that are known as strands. Now which are right-handed coiled, which are right-handed coil, antiparallel. These right-handed coils are antiparallel, and that is one strand runs in five to three direction and the another strand runs in and the another strand runs in 3 to 5 direction. So it was about the right-handed coil, that is antiparallel. Now the complementary, and this is complementary to each other and is complementary to each other. Now they are held together by hydrogen bonds established between the bases of pairs. So these two strands are held together by hydrogen bonds established between bonds established between the bases of pairs. So these strands are held together by the hydrogen bonds that are established between the bases of the pairs. Now the next point is base pairing, that is for that hydrogen bonds. It occurs among adenine, that is A, thymine, that is T, cytosine, cytosine, that is C, and guanine, that is G. Now the next one is two hydrogen bonds are formed. Two hydrogen bonds are formed between A and T, that is adenine and thymine, while 3 between cytosine and guanine. So two hydrogen bonds are formed between A and T, that is adenine and thymine, and 3 between, and 3 bonds are formed between C and G, that is cytosine and guanine. Now, Watson and Crick model of DNA. Now, Watson and Crick model of DNA describes that first one is each nucleotide pair needs pairs need 3.4 strand space of double helix It was the first statement. Now the second one is one complete turn of helix. One complete turn of helix has of 10 nucleotide pairs. Nucleotide pairs occupying 34 strand space. That because this 3.4 into 10, this 3.4 into 10, that is equals to 34 space. Now the third one is the diameter of DNA molecules. The diameter of DNA molecules is approximately 20 angstrom. Now the next statement is there is a fixed distance of fixed distance of fixed distance of 10.08 between the two sugar moieties in opposite strands. Between the two between the two sugar moieties in opposite strands. Thus, only certain base pairs can fit into the structure. Thus, only certain base pairs can fit into the structure. That means not all base pairs can fit in the DNA structure. Only certain base pairs can fit into the structure in which the fixed distance of 10.08 angstrom between two sugar moieties in opposite strands. Now, the last one is if any long base pair sequence exists at many different places. Base pair sequence exists at many different places throughout the DNA. It is called repetitive or repetitious DNA. It is called repetitive DNA. The highly repetitive DNA is known as satellite DNA. Now, the highly repetitive DNA is called satellite DNA. So, these are the structure of Watson and Crick model of DNA. And I have discussed the formation of DNA before. Now, you can pause my video and watch it and repeat it as you need.